Thanks for giving me the opportunity to speak at that really wonderful meeting. He told you already I will speak today not just on an affair of the heart and show you the effect of Papaverine on the cardiac sodium channel 1.5. Let me shortly introduce who we are. We are a consortium, a research consortium of sodium channel scientists in Aachen. And in our consortium we have geneticists, electrophysiologists, physiologists, pharmacologists like me, and computer modelers, and we are interested in sodium channel related pain disorders from molecular mechanisms towards personalized treatment. And together we are the so-called Sodium Channel Network Aachen, and very recently Angelika Lampel and me have founded the Scientific Center for Neuropathic Pain in Aachen. If you are interested, please visit our homepage, what we are doing. Okay, I will start with an affair of the heart, but hopefully at the end of the talk I will have convinced you that this has something to do with sodium channels and electrophysiology. And this came up with a collaboration partner at the Cardiovascular Center in Aalst. This is located in Belgium, and it was Bernard de Bruyne and Monica Colebuina. And first, I should mention what's about the disease. Most of you will know that coronary artery disease is one main cause for morbidity and mortality in high-income countries. And usually if you have a patient who is suspected for coronary artery disease, these patients, when they have a high likelihood of the disease, they undergo cardiac catheterization, performing angiography like it is shown here. And in most cases, or very often, you find then a stenosis. Here's the coronary artery, and you see a stenosis. And then the cardiologists have to decide whether an intervention, the so-called percutaneous coronary intervention, mostly in form of a stent implantation, is beneficial for the patient. And this decision can be made by the so-called fractional flow reserve measurement, FFR in short, and this consists of measuring the pressure after the stenosis and measuring the pressure before the stenosis and comparing both values. And then you can decide whether stenting this lesion may be beneficial for the patient or not. There are other methods, but this is the most common method, and the group from Bernard in Aalst, they published 5 years ago, they published the 5-year outcome of their study. And as you can see here, there was shown that coronary intervention guided by the fractional flow reserve measurement was beneficial. You can see the composite primary endpoint was death or myocardial infarction, and it was less common in the PCI treated people as compared to the ones with medical therapy alone. Yeah, and the thing is, when you perform these FFR measurements, you need to induce maximal hyperemia in the coronary artery of interest. And this is done in high-income countries who have lots of money. This is mostly done by adenosine infusion. Some people are also using regadenosone. Then you have the adenosine 2A receptor mediated vasodilation in the coronary artery. However, this treatment is relatively uncomfortable for the patient. They feel a heat sensation, flushing, and some of them also exhibit dyspnea. And the compound, or the duration of action is very short, and the drug is very expensive. Another possibility is to use Papaverine, and you know this is a very old drug. It's from the opium poppy, but it has no morphine-like effects, pharmacologically speaking, but it enables maximal hyperemia in the coronary artery, and this is injected in the coronary artery via the cardiac catheter. This is much more comfortable for the patient. The duration of action lasts for the full FFR measurement. It's very inexpensive, thus available also in most countries of the world. However, there's a big problem, and that's the risk of malignant ventricular tachycardia VTA in form of ventricular tachycardia, ventricular fibrillation, or TDP arrhythmia. As you can see also here in these two case reports, that's not totally uncommon with this drug. And then, the colleagues from the Cardiovascular Center in Aalst, they did a really large prospective single-center, single-arm, all-comer study, and they included 2,150 patients, and all of these patients got Papaverine for FFR measurement during cardiac catheterization. And from these more than 2,000 patients, 24 patients, this is roughly 1.1% of this population, exhibited ventricular tachyarrhythmia, and then we came into play. The clinicians asked what may be the mechanism why Papaverine may induce this arrhythmia. Thus, we went from bedside to bench and thought, maybe as an ion channel scientist, maybe the channels which are responsible for the heart rhythm are the mechanism. And then we looked into literature, and you can find some reports that Papaverine blocks the Herc channel pore, and there you find also individual reports of this tachyarrhythmia, which may be due to the Herc channel. If you look for the sodium channel 1.5 or the T-type calcium channel, the effects on the cardiac rhythm is largely unclear after Papaverine induction. And as a pharmacologist, then we went on and did concentration response curves of the sodium channel 1.5 and the Herc channel by using automatic patch clamp. And as you can see here, for the sodium channel on the left and for the Herc channel on the right, Papaverine, here shown in blue, shows a block of the sodium channel 1.5 with a micromolar potency of an IC50 of around 400 micromolar. And as a positive control, we used maxilitine, and there we found an IC50 value of 50 micromolar, which is very close to the value known from literature. If we look to the Herc channel, and that was known from literature before that this channel is blocked by Papaverine, we have an IC50 value of around 6 micromolar. For amitriptyline as a positive control, we estimated a value of 4.5 micromolar. Okay, let's have a look what we have already and what may be the open questions. We could already show that Papaverine blocks the sodium channel 1.5 with low micromolar potency, blocks the Herc channel with a higher micromolar potency, and then you have to relate it to the concentration used in the cardiac catheterization procedure. Then you can see 10 to 20 millimolar is the concentration they inject into the coronary artery. However, we do not know the target engagement, how much of the drug will reach the sodium channels, for instance, expressed in the cardiomyocytes. So far, the results that these ion channels are blocked may explain the effect of Papaverine on the heart rhythm. However, they do not explain why only 1% of the patients develop ventricular tachyarrhythmia after getting this drug. And then we speculated whether genetic variations associated with cardiac arrhythmias may play a role, and then we went for full exome sequencing of 21 of the 24 patients who developed the VTA in response of the Papaverine treatment. And here I show you these results of full exome sequencing, and the sodium channels I will show you on the next slide. There were found in 7 patients mutations in genes associated with cardiac arrhythmias. There was 1 Herc channel mutation that was, according to the ClinVar database, associated with long QT syndrome or arrhythmia. Myosin heavy chain mutations, ryanodine receptor, placophyllene, which is no interaction partner of some cardiac ion channels, and so on. At the ion channel meeting, I should focus on the ion channels. I think the Herc story is going in another lab, but we have focused on the sodium channel mutants, and these are shown here. We found 4 different variants of uncertain significance for the Brugada syndrome in the SCN5A gene encoding the cardiac sodium channel 1.5. These are shown here. And 2 of these had a very rare frequency. In the normal population, 1 was more common, and the T1022A shows an unknown frequency. What's the Brugada syndrome? I think most of you will know at this meeting. Brugada syndrome is a cardiac condition, mainly due to inherited malfunction of the sodium channel, the Brugada syndrome 1, and showing a typical ECG phenotype shown here. And it was first described by the Brugada brothers, Pedro Brugada and his brother Joseph. And interestingly, Pedro Brugada worked at the cardiovascular center in Aalst till 2006. Then the circle is closed at this center. What did we do next? We went for NAF 1.5 biophysics and pharmacology. For that we used HEX cells stably expressing the sodium channel and its variants, and these were generated by the Flevin system. I think most of you will also know. And then we went for automated high throughput pledge clamp using the cube. And yeah, next I can show you what we do in these protocols. You can see that we not go for a single voltage clamp protocol, we go for a really set of voltage clamp protocols, monitor pulses, IV activation, inactivation curves, recovery from inactivation, trains, REMs, cardiac action potential like voltage clamp protocols, and so on. And these are run first in the absence of Papaverine, and then the same protocols were run on the same cells in presence of different concentrations of Papaverine. And on the next slide I show you the results. There were lots of biophysical features that were indistinguishable between the wild type and the variant receptors. But if you look here to the activation and fast inactivation curves, you can see that there's a change in the inactivation curve. There's no large difference between the variants and the wild type. But for the activation curve, you see especially for this mutant T1022 to alanine, you see a depolarizing shift of the activation curve. And when you look to the same measurements in presence of the drug, this is a concentration which is perfectly water soluble. This can be done up to 140 micromolar. And then you can see that the depolarizing shift is even more pronounced in presence of the drug. The people with the sharp eyes of you will also have seen that we have a shift of the inactivation curve, but that was only time dependent. It was also present in the saline control, and thus this may be not due to the Papaverine treatment. Then there was a difference in the recovery from inactivation. On the left you see the curves without Papaverine, and you see wild type and mu order variants are not distinguishable. And also in the inset here showing the first 200 milliseconds to see the fast component a little bit better. There was roughly no difference. And the picture is different when we look in the presence of 100 micromolar of the drug. Then you can see, maybe best here in this inset, you can see that the fast end as well as the slow component was a little bit delayed, but especially for the mutant 1022A we saw a very marked delay of the recovery from inactivation curve, and this is also shown here in the statistics. You see clearly, you see a trend for a delay here, the time constants, the fast time constant and the slow time constant from by exponential curve fitting. You see there's clearly a trend that this is the recovery is delayed in presence of the drug, but there was no statistically significant difference, with exception of this mutant, the T1022A shows a significantly delayed recovery from inactivation as well, for the fast and for the slow component. Yeah, so far I could show that 3 of these 4 variants have biophysical properties very similar to the sodium channel wild type, but the 1022 alanine shows a loss of function phenotype, which is characterized by a depolarizing shift in activation, which is much more pronounced in presence of papaverine, and we see a delayed recovery from inactivation in presence of papaverine. And then Angelika and me, we thought whether this phenotype is compatible with a papaverine-induced pro-arrhythmic phenotype, and the answer is yes. And I brought you here a paper from the Brugada brothers from 1999, and there was also a mutant of the Brugada syndrome in the sodium channel 1.5, and you can see here unmasked by measurements at a higher, more physiological temperature, they saw a delayed recovery from inactivation, and they saw a depolarizing shift in the activation time curves. This is also similar when you look into other publications, but here it's interesting to see that the so far known variants of this NAF 1.5 associated with the Brugada syndrome, they are spread all over the receptor. There's no special focus, and then we have some additional mutation there. I thought I will give you some structural considerations or an idea how the mechanism of this biophysical change may be explainable, and then we look to the paper from the Carroll group from 2021, 2020, the one also, and also the with the human structures from the Yarn lab from Princeton. But then we recognized that the intracellular domains and the distal C term were partially delayed for the generation of these structures, or were without sufficient resolution. Then we tried AlphaFold, but to be honest, these pictures I do not show you because they are really, that's a bad prediction of the intracellular domains. Thus, I will show you only the cartoon from a colleague from Switzerland, and showing here the positions of the mutants or the variants that we have analyzed, and the most strong effect was the 1022 to alanine mutation in the loop of in the linker between domain 2 and 3, and this is a region where, for instance, the ankyrin binding motif for the cytoskeleton proteins is localized, or other interacting proteins. Means the mechanism is so far unclear how this happens, and concluding is we found this loss of function phenotype, which is facilitated by papaverine. It's not only one additional Brugada syndrome mutation, it's we found that special biophysical features are only changed after treatment with this drug. At the end, we have to say that in only one of the 22 patients who went for sequencing, the variant may have the sodium channel 1.5 variant may have caused the VTA, and but in the end this may suggest that variant-specific druggability may be successful in selected variants. And one thing which is currently under study is to do the measurements at more physiological temperatures. This is very important, as you have seen in the Brugada paper, and we are able to do these measurements at 30 degrees, 35 degrees with very high efficiency. We have more than 85% efficiency by using our system, and there we will see, hopefully, some additional changes. And also we go for molecular modeling and molecular dynamic simulation to explain why these mutants may cause these biophysical changes. Let me shortly show you this outlook. I mentioned the variant-specific druggability, and this is also of interest in our labs, in Angelika's and my lab. And beside the wet lab screening with the normal drug screening and also virtual screening by a colleague from Forschungszentrum Jülich, we went another approach. It's the so-called precision to treat project funded from the federal ministry. And we have in Aachen, we have a very large cohort of neuropathic pain patients, more than 300 people, and all of them are sequenced fully. We have full exome sequencing of all these patients, and we got money to have induced pluripotent stem cells of 20 of these patients. These cells are then differentiated to patient sensory neurons, and thus they will contain all the variants of the ion channels the patient has. And these cells then went to the APC electrophysiology, and we used the Pharmacon library, which consists of all approved drugs on the market, and then hopefully we will find some variant-specific druggability or something which can be used in later years for personalized drug treatment. Okay, at the end I would like to thank the collaboration partners, especially the people from Cardiovascular Center in Aachen, the funding agency, and we, the CUBE team in Aachen, thanks you for your attention. Thank you very much.
Well, thank you, Ralf, for a great talk. Let's open up for questions. Yes, at the back.
Hi, great talk. Can I ask if you looked at affinity at the late current, because the low rate, the slow rate of dissociation suggests that's what it's hitting, and does that variant have a higher late current than the other?
Yeah, that's also a common finding is indeed in the Brugada syndrome that the late current is increased, and to be honest, the analysis of the curve fitting was not available when I prepared for the talk, but for sure this is of interest. And also the current density. But there I should add, it's not totally easy in this heterologous overexpression system to get some results for current densities. It's not straightforward, but we have it onto the screen, yeah.
Question
Thanks for the nice talk. Just wondering, because I also test the Papaverine on the calcium channel, and it seems also to inhibit the calcium pathway, right?
Maybe possible, but we did not analyze the calcium channel. We did mainly focus on the HERG and on the sodium channel 1.5, but for sure it's interesting to have a look to the calcium channel.
Okay, because I have some feedback from the review at that time that when I was speaking about the calcium, he said, what happened with the sodium? So
yeah, I think I told you before, we should come together. That's clear.
All right. Let's thank Ralf again. Thank you.